This call is now being recorded.
Can you see the screen, children? Just unmute and tell one of you whether it is visible, the screen.
Yes, ma'am.
Yeah.
Yes, ma'am.
Yes, ma'am.
Okay. Thank you. Now, let us start with the next part of this chapter, that is pleiotropy. So, what all these examples we are discussing, these are the deviations from the Mendel's laws we were talking. We discussed about the incomplete dominance, codominance, then multiple alleles. Now this is the next example which we are talking, next part of this chapter, that is pleiotropy. Now what does it mean by, I hope your textbook is ready with you, pleiotropy, page number 55. When a single gene controls two or more different traits, it is called as pleiotropic gene, and the phenomenon is called pleiotropy or pleiotropism. Now, in this case of pleiotropy, the ratio is 1:2 instead of 3:1. Now, why and how? I will tell you with the help of these slides. Okay, now pleiotropy, what does it mean? Multiple effects of a single gene. A specific gene controls a specific phenotypic trait. That means this is a single gene, and in pleiotropy, it is showing all these different traits. So multiple traits. So examples of pleiotropy are many more, but before going to all these, first we are going to start with one example where a single gene influences more than one phenotypic trait, that is the gene expression. And the best example which is given in your textbook is sickle cell anemia. Try to understand what exactly it is. Sickle cell, sickle means what? And see, these are the blood cells. These are the RBCs, red blood cells. The normal size and shape of the blood cells is like this, spherical. Okay, little bit rounded, spherical. This is the normal RBC. And when it becomes sickle shape, then this is the shape. That is called as half moon shape. Okay, like half moon. Yes. So this is what happens. When it happens, this is because of the autosomal recessive genetic defect. That is the blood disorder. RBCs are characterized by an abnormal, rigid sickle shape in hypoxia condition. What does it mean, hypoxia? Hypo means below. Oxygen level when is below than the normal, in that condition, the cells which are normal in this disc shape or rounded disc-like shape, they become sickle shaped. And then what happens because of this? This is, what is the hypoxia condition? It occurs at high altitude or under physical stress. This happens when at high altitude or under physical stress. Then what happens when the blood cells change their shape from normal, round to the sickle? Then what happens then? These cells get clumped and clogged in the small blood vessels. Then because of the change in the shape of the blood cells, RBCs, they may get clogged at one place in the blood vessels. And because of what? That particular organ may damage. Then physical weakness, pain, and that may result in paralytic condition also. Okay, and sickle cell anemia is controlled by a single pair of alleles, and that is present on chromosome number 11. HbA, which is considered to be the normal hemoglobin, and the HBS, which is representing HB for hemoglobin, that is sickle cell. Now, the chromosome number 11, where this gene is present, homozygous individuals for sickle cell anemia, they are considered to be represented as HBS and HBS. Just now we were talking about the recessive condition. When this phenomena or this condition of anemia is observed, when the gene is in the homozygous recessive form, that means both the alleles are showing it in the recessive form, nature. That is, then only this disease is expressed, or this type of anemia is to be observed. But if the individual is heterozygous, that means what? One is normal and the other is sickle cell, then it is represented as HBA and HBS. But here in this condition, heterozygous form, as the normal gene, that allele is dominant over the sickle cell, so that this person phenotypically is normal. It just carries this gene for the sickle cell, and that's why this is heterozygous condition. Okay, now what you need to remember in this is, even though two sickle cell alleles are necessary to cause sickle cell anemia, one dose also can affect the phenotype. Person heterozygous to sickle cell trait can usually lead a healthy life, as I told you just now. Sickle cell anemia is caused by a point mutation in the DNA that codes for the beta globin polypeptide chains of the hemoglobin molecule. Hemoglobin molecule, where it is present, you know that hemoglobin is present in the RBC's blood, and this hemoglobin is made up of 4 chains, 2 alpha chains and 2 beta chains. Again, that structure I will explain in another slide. But remember, because of the defect in one of the beta chains, it is observed. It causes the replacement of the glutamic acid in 6th position by valine. In one of the beta chains, this normal beta chain position is at 6th place. That is replacement of glutamic acid in the 6th position by valine. Glutamic acid is present at the 6th position, which is replaced by valine, and because of this small change, and that occurs because of mutation, because of such small change, that oxygen carrying capacity of the hemoglobin is reduced, and in prolonged periods of reduced oxygen content in the blood, the heterozygous person may suffer from symptoms of this SCD, sickle cell disease, as both normal and sickle cell hemoglobin are formed in them. Right? So if it is prolonged, for the prolonged period, then this normal heterozygous person also may feel such condition as sickle cell person. So what does this phenomena mean? Due to codominance of allele, it may happen. The heterozygous individuals for sickle cell anemia are resistant to malaria, such as falciparum malaria. This is just additional point. Now you can see what happens here. This is the blood cell, where these are the normal blood cells. They are passing very easily through the blood vessels, these RBCs, red blood cells, as they are spherical, rounded in shape. Very easily they can be passed when they are in the normal condition. But when these cells are changed in the shape to sickle shaped, then what happens? They make it clumped, and then there are chances of getting that blood vessel blocked, clogged, because of the change in the shape of these blood vessels. And then this condition may result in the severe form to paralytic form. Also, paralysis also may take place. Some organs may damage. If it is there observed in the brain, then again hemorrhage. So such type of conditions are observed. Now this sickle cell anemia, which is where in an inherited red blood cell disorder, these normal blood cells are round like donuts, and they move through small blood tubes in the body and deliver oxygen. But in the sickle shaped form, there are chances of getting those blood vessels clogged by these RBCs, that means sickle shaped cells, and it may damage that particular organ where the blood vessel is. Now, how this phenomena is observed in case of parents, how it takes place, which is given in your textbook. Just see figure 3.4. What are the phenotypes in case of parents? Both the parents when are carrier, then what happens? So genotypes of the carrier person with, that is the heterozygous condition, is what? HBA, HBS for both the parents. Then these carriers, they are producing two types of gametes. That means two types of gametes are observed here. One is with HBA normal and the other as HBS, that is the sickle shape. Again, here also two types of gametes are formed. Then the, in their offsprings, what are the results or the genotypes of the offsprings produced? You can see this normal gamete, when it is fused with the normal gamete of the another, then normal person, normal child with both the genes normal, that means totally normal offspring is produced. Then when this gamete joins with the sickle celled gamete, then there are the chances, that is when the offspring which is produced is the carrier. Again the same, one more offspring of the same type, that means two carriers are produced. And then the third one, that is totally when HBS gamete fuses with the HBS gamete, then there the person or the offspring which is produced is having both the genes in the recessive condition, that means HBS, that means this offspring is sickle cell which carries this disease, that means sickle cell anemia, cannot live for a longer life, and that offspring will die in the early stages within few months. Or so that's why this is considered as the sickle cell ratio. In case of sickle cell anemia, the normal 1:2:1 ratio is not observed, or the genotypic ratio as 3:1 as per the Mendel slots is not observed. But here, as this homozygous recessive person offspring dies, only how many remains? Only three. One is totally completely normal offspring and the two carriers. So instead of 3:1, the ratio is observed as 1:2 and normal is to carrier. So this is what you have to remember in this case as sickle cell anemia, which is the example of pleiotropy. Now, for what else this is example, that also you need to remember. Now, when it will come, I will tell you. Now, we'll talk about the structure of chromosome first, and then we'll observe, and then we'll study what type of inheritance it is seen and all the details. Okay, what does chromosome means? Chroma means body. In Greek, it is called as chroma. Chroma means color and soma means body. Chroma is color, sorry. Chroma is color and soma means body. So, the colored body, which is stained by a particular dye or the stain. That is, German scientists Schleiden, Virchow and Buchli were among the first scientists who recognized the structures which now are familiar as chromosomes. Now, few basic things you should know about the chromosomes. That is, DNA molecule with part or all of the genetic material that is considered a genome of an organism. It is. During cell division, the nucleus of the cells in the nucleoplasma have a kind of yarn type, that means a thread-like structure. These formative organisms in the nucleus are called chromosomes. Thread-like molecules that carry hereditary information for everything from height to eye color. These are nothing but the responsible one as chromosome. They are made up of protein and one molecule of deoxyribonucleic acid. So they are made up of proteins and DNA, deoxyribonucleic acids. Two types of proteins, histone protein and non-histone proteins, again as per the type of species, but in general you need to remember they are made up of protein and nucleic acid as DNA. They are visible under light microscope when? Visible under light microscope only when the cell is undergoing the cell division, and that too in the metaphase stage. It is clearly observed because of the condensed form. Okay, now last year you have learned all the stages of mitosis. If you remember, mitosis, cell division, a type of cell division, there you have learned prophase, metaphase, anaphase, telophase, etc. So that metaphase stage only it is observable under light microscope only during the cell division, and that too only in the metaphase phase stage of cell division. These chromosomes are visible. They are made up of protein and one molecule of DNA, which contains an organism's genetic instructions and passed down from parents to their offsprings. When we talk about the human beings, their number is also fixed for the number of chromosomes is fixed for that particular species. Okay, and most chromosomes are arranged in pairs within the nucleus of a cell. Humans have 22 these chromosome pairs called as autosomes and only one pair of sex chromosomes. So total 23. Now we'll see where it is present. This is the cell. At the center of the cell, nucleus is present, and in the nucleus these chromosomes are present, and on this chromosome, that chromosome which is made up of DNA and protein. This is the structure of DNA that we have already learned to some extent last year also, and we will study in detail this year also. Now, what are the parts of the chromosome? That chromosome shows all these parts as chromonemata, centromere, or this is also called as primary constriction, chromomere, pellicle, matrix, secondary constriction, and satellite. One by one, we will see now. What are these parts? Embedded in the matrix of each chromosome are two identical spirally coiled threads. They are called as chromonemata. Chromonema is a singular, chromonemata is a plural. So there are two identical spirally coiled threads. Okay. At metaphase, each chromosome consists of two symmetrical structures, the chromatids, each of which contains a single DNA molecule. The chromatids are attached to each other only by the centromere and become separated at the start of the anaphase when sister chromatids migrate to opposite poles. And you have learned these stages, so I will not explain right now, but we'll try to study the structure of chromosomes. Now, under lighter staining region, a lighter staining region appears as a constriction or thinner segment of the chromosome and usually called as centromere or primary constriction. I will show in the figure also. And according to the presence of the centromere in the chromosome, these chromosomes are known by these different names. Means they are called as metacentric, submetacentric, acrocentric, and telocentric. So I'll show you in this diagram. First, you see this diagram, which is considered to be this one, metacentric. Metacentric chromosome means when the centromere is present at the center of the chromosome, and these are the two chromatids attached to each other at the point that is called as centromere or primary constriction. And this centromere, when it's present at the center, making the arms equal, which gives V shape. In V later, both the arms are of equal length, right? So that's why this metacentric chromosome may appear as V-shaped, okay, because the arms are in equal length. When the centromere is situated at the center, the arms of the chromosomes are of equal length, giving V shape. So that's why this is called as metacentric. Now, submetacentric means this centromere is little bit away from the center of the total length of the chromosome, and it forms L shape. In L letter, vertical and horizontal. Vertical longer than the horizontal, so it gives L shape. So L shaped chromosomes, or they are called as metacentric chromosomes, where the centromere is little bit away from the center, making the arms unequal. Then next is what? Acrocentric. In the acrocentric condition, the centromere is present at one end, making very unequal length of the chromosomes, where one arm is very short and the other is very long. So this may appear as J shape in alphabets, J letter. How it looks, that J, one is very short arm, very small short arm, and the other is long. So like J shape, it gives the appearance as, and this is what called as acrocentric. And the last one, that is telocentric, where there is no arm, short arm, but this centromere is present at the end of the chromosome. So you can see only one arm, longer one. There is no short arm as such seen here. So at the terminal end it is present. So this is called as the telocentric chromosome. So depending on the position of the centromere, these chromosomes are called as metacentric, submetacentric, acrocentric, and telocentric. Centromere is nothing but the point of attachment of these two chromatids. Now, see this in this diagram is again very clear. The chromosomes, particularly in early meiotic prophase, were differentiated into morphologically distinct regions which are constant in size and position, and these regions are called as chromomere. Now, see, this is the metacentric chromosome. This is one chromatid. Now you can see only like this, but when you observe in the metaphase stage, it gives the appearance of X shape. Here is the centromere, or called as kinetochore, or the secondary constriction, primary constriction, where that is at that point these chromonema are attached, chromatids are attached to each other. Okay, so this is the each chromatid, you can see there's the presence of chromomere, where these are spirally arranged chromonema present, right? So don't get confused between these terminologies. Try to understand, though not immediately, as and when you read it again and again, you will understand. See, this is the simple form, metaphasic stage of chromosome, where it gives the appearance of X later, where at the center, this is the centromere, and these are the arms. So these are the chromatids. Now, what does it mean by pellicle? That means the boundary of the chromosome. You can consider this as a boundary of the chromosome is called, that is the bounded, it is that chromosome is, each chromosome is bounded by a membrane called pellicle. It's very thin and formed of achromatic substance, that means without any color. This membrane encloses a jelly-like substance which is usually called matrix. So inside the pellicle, that means that boundary, that membrane, whatever the jelly-like substance is present, that is called as matrix. And the matrix is present the chromonemata. The matrix is formed of achromatic or non-genetic material. It is not made up of genetic material, but it is non-genetic, and achromatic means it's colorless. The function and structure of matrix is not known as yet, but it is assumed. It aids in keeping the chromonemata within boundaries. That means, but that's why boundaries are made within the bounds, so that the maneuver of the chromosome during cell division can take place unhindered. It may also serve as an insulating sheet for the genes during cell division. So these are the parts of the chromosome. Now remaining few additional parts also you should know that this is the primary constriction. Now this is one more constriction in the cells. It is constricted at that particular point. You can see the shape when you see this is the this diameter at this point, and the diameter of the chromosome at this part. Here it is constricted. So this is the second constriction. So this is called a secondary constriction. So secondary constriction mark location at which the nucleoli are assembled. Now, when you study the structure of nucleus, and if you remember it in detail last year, what is present in the nucleus? One or two or many nucleoli are present, right? So, nucleoli, wherever the secondary constriction is present, that marks that the presence of nucleoli there. That means the location of the nucleoli is marked by the presence of this secondary constriction over there in the chromosomes. The chromosomes, in addition to primary constriction, that means centromere, possess secondary constriction at any point of the chromosome. And it's often associated with the nucleolus during the interphase and may take part in the reorganization of the nucleolus at the end of the cell division. If you remember the cell cycle, in the cell cycle, two phases, important phases we have learned, that is one is interphase and the other is cell division phase. In the cell division phase, that is mitotic division, we have learned there are different stages as prophase, metaphase, anaphase and telophase come. And the another, when this is completed, what is the phase? That is the interphase. So in the interphase, the position of the secondary constriction is seen, whereas during the cell division, the constriction, that is secondary constriction, may change its position. So that is what the role of secondary constriction. That is what reorganization of the nucleolus, it may take place. Now, what are the satellite bodies? Because of the secondary constriction, the part of the chromosome which is observed as a small part at one of the ends of the chromosome, and that smaller part, sometimes it is observable, sometimes it is very small, which gives an appearance of a satellite. So that's why this is called also called a satellite body. So the part of the chromosome which is present beyond secondary constriction is called as satellite body or trabant. It varies in shape according to the position of secondary constriction. If secondary constriction is very close to the end of the chromosome, the satellite may be barely perceptible dot. Just you can see a dot also. If it is that position of the secondary constriction is at the end of the chromosome, very close to the end, then it may appear as a dot also, that satellite. So there are, depending on the position of the secondary constrictions, satellite bodies are observed. They are visible properly or sometimes as just a dot. And depending on the number also, the number of secondary constrictions, this number of satellite bodies are seen. Now this is what given in your textbook, this diagram very clear. This is the A, where metaphasic stage of chromosome, where this is the centromere, these are the arms, and these are the chromonemata, and this is the matrix, and the boundary of this chromosome is called as pellicle. And in the matrix, these chromonemata are present. And when you see a single one arm where this is the primary constriction, which is also called as centromere or also kinetochore. It is also known as kinetochore. Then primary constriction, and then this is the secondary constriction. Because of secondary constriction, whatever the smaller body at the end of the chromosome, you can see that is called as satellite body. And because of the presence of the secondary constriction, it is also called as nucleolar organizer, because this only takes place in the reorganization of nucleoli. That's why this secondary constriction is also called as nucleolar organizer. Okay, and here is the location of the nucleolus. So that's why this is As I told you, the presence of secondary construction locates is responsible for knowing the presence of nucleoli or nucleolus. These are the chromomeres, spiral shaped structures you can see, and the end of the chromosome beyond the secondary construction, that is called as telomere. That is the terminal end. So these are all the parts of the chromosome structure. Now we'll talk about the number of chromosome, that is how many number of chromosomes are present, and accordingly that depending on the number of chromosomes, the species shows. That means each and every species is having a particular number of chromosomes. Now that also, it is when it is fixed to that particular species, sometimes because of different reasons, what are the reasons that we'll discuss later, there may be change in the number of chromosomes also. So we'll talk about first what does it mean by ploidy, euploidy and uneuploidy, and then we'll talk about the further details. The number of chromosomes is specific and constant for a particular species. For example, human beings have total 23 pairs, that means 46 total, out of which 44 are autosomes, 2 are sex chromosomes, like that. For that particular species, it is fixed. It is, and it remains constant also for that particular species. So, therefore, it is of great importance in the study of phylogeny and taxonomy of the species. Phylogeny is the branch of biology where we study the you can say the history of that particular part. In detail, we can study, we can know. And then taxonomy, that is the systematic classification is possible, where we can group, we can place that particular species in the classification. Okay, so the term ploidy speaks for the degree of repetition of the primary basic number of the chromosome. Now, primary basic number, when is considered to be X, that means it is when in the haploid condition, it is represented as X is equal to N. N means haploid condition, only one set. N represents the number. Number means only one set of chromosome. Okay, so when the chromosome number in a cell is the exact multiple of the primary basic number, then it is called as euploidy. Means if it is N, 2N, 3N, 4N, like that, means when it is in the multiples of the primary basic number, when we are considering the basic number as N or X, then 2X or 3X or 4X. And if X is equal to N, that means if the haploid only one set, then it is considered to be haploid. So when the number of chromosome is in a cell is exact multiple of the basic primary number, then it is called as euploidy. And euploids include monoploidy, or this is also called as a haploid state. That means with only one set of chromosomes where X is equal to N. When two sets, then it is called as diploid. That means two sets of chromosomes. When three sets of chromosomes, that is called as triploidy. Tetra means 4, and so on. And when the chromosome number is not in the exact multiple of the haploid set, then it is described as uneuploidy. When it is not in the multiple of haploid set, either it is more or less, then it is not equal. It is not in multiple of the haploid set, exact multiples of the haploid set, then it is called as uneuploidy. Uneuploidy, that is unequal. It becomes, it is either addition or deletion or of one or more chromosomes to the total number of chromosomes. So that's why in this chart it is very clear. Now you try to understand. Ploidy is of two types, euploidy and uneuploidy. Euploidy means the base, when the number is exactly in the multiples of the basic primary number. When only one, monoploidy. Then two sets of chromosomes, diploidy. Polyploidy means 3 sets, 4 sets, and means exactly in the multiple of that basic primary basic number. So that is called as euploidy. Now Aneuploidy means it is not equal. It is not in the multiple. Then it is again divided as hypoploidy and hyper. Hypo means below. When the number is below the ploidy, that is the multiples, then that is called as hypo. And hyper means above. When that ploidy, that means it is more than the euploidy, that means the multiples of that exact multiples more than that, then that is called as hyperploidy. Then hypoploidy can be monosomy, when mono means single, one chromosome is less than the normal. Nullisomy when 2n minus 2. Trisomy 2n plus. Now trisomy is the example of hyperploidy, and tetrasomy, that is 2n plus 2. So these are the examples of hyperploidy and hypoploidy. Okay. So now the next what we are going to study is sex determination in the organisms. Now we'll talk about the next point, that is sex determination. There are different types in human beings, in birds, in insects. Different ways of determination in the sex is observed. Now one by one we'll talk. That is XXXY type, which is observed in human beings. Now XXXY type of male, that is heterogametic sex determination. Now in case of human beings and some insects such as Drosophila, where heterogametic and homogametic, these are the two stages. Homo means similar. When gametes are similar, that condition is called as homogametic. When gametes are different, then that condition is called as heterogametic. Now in case of human beings and few insects like Drosophila, males are heterogametic, whereas females are homogametic. That means gametes in females are similar, and in case of human beings and some insects, the gametes in males are different. Like in case of females, both sex chromosomes are XX type, whereas in males, one is X and the other is Y. So this is what called as heterogametic condition, or XXXY type of heterogametic sex determination in case of human beings. So males are heterogametic and females are homogametic, because males have X and Y, two different types of chromosomes. They produce two types of sperms. Females are homogametic as they produce only one type of ova, similar, that is X chromosome each. Now, the sex of the offspring depends on the fertilizing sperm of X or Y type. Now, how it takes place? Now, these are the two types of sperms, X and Y, and ova produce eggs are of similar XX. Then, when this X fuses with the X, the result is female child. Again, this X when fuses with the another X, the result is female child. But when this ovum fuses with the Y type of sperm, then that is XY. Same when this gamete, that is the egg ovum, fuses with the Y type of sperm, then that is XY. That means the offspring which is produced is male. So this is what called as heterogametic sex determination in human beings, where males are determining the sex of the offspring or the child in case of human beings. But in now this another case in case of birds and even in some reptiles and in some other species also, females are heterogametic. In case of human beings, males are heterogametic. Now in case of birds, see, the females are heterogametic. Who decides the sex of the offspring in birds? By whether it is boy or a girl, whether it is male or the female. This system is called as ZWZZ sex determination. In case of human beings, it is XXXY. In case of birds, it is ZWZZ. ZWZZ. That is because the females are heterogametic, which are represented as ZW, while males are homogametic, represented as ZZ. Here in case of birds, these allosomes, two dissimilar gametes are AA, that means autosomes, and ZW. Females produce ova with Z chromosome and ova with W chromosomes. So males are homogametic and females are heterogametic in case of birds. Males, they produce sperms with only one type, similar gametes, that is Z chromosomes. So one half of the ova are with allosome as Z, and other half ova are with allosome as W. So, but all sperms have allosomes as Z. So females produce two kinds of ova, whereas males produce similar type of sperms. That is, the sex of the offspring depends on the type of the ovum that is fertilized. Now, when here female gametes are represented as ZW and the male as ZZ. So the resultant of this, when this ovum matches with the Z, the resultant is Z, and when the ovum with the chromosome W fuses with the Z chromosome of male, then the resultant is ZW. That means accordingly, female and male. This is the sign of female, which it represents, and this is the sign of the male. That means two males, two females, that offsprings in case of birds. So in case of birds, female is heterogametic. In case of human beings, males are heterogametic. Now one more type of category is there, that is called as haplodiploidy, and that is observed in honeybees. What does it mean? Try to understand. Mechanism of sex determination in honeybees, ants and wasps is called as hymenopteran insects. That means these are all hymenopteran insects. That means this mechanism, that is the sex of the offspring, is determined by the number of sets of chromosomes it receives. According to how many sets of chromosomes it receives, then the sex of the offspring is determined. Now how it is, try to understand. Haplodiploid type of sex determination, this state, fertilized eggs developed into females having double the number of chromosomes. This is the honeybee, which is diploid. That means two sets of chromosomes are present, double the number of chromosomes. Two sets of chromosomes are present. Okay. Unfertilized eggs developed into males having half the number of chromosomes. And which are they called? They are called as parthenogenic eggs. The fertilized eggs are with two sets of chromosomes, right? Whereas unfertilized are having with only one set. That means they are half the number of chromosomes, which are called as parthenogenic eggs. Unfertilized eggs develop into males having half the number of chromosomes. And what is that state called as? Hence this method is named as haplodiploidy. Haploid in case of males and diploid in case of females. Try to understand. So that's why this is called as haplodiploid in honeybees. Now, drones contain 16 chromosomes. So this state is called as haploid. In Apis mellifera, that means in honeybees, the number of chromosomes, drones, they are called as males. Drones is the name word in case of honeybees for males. They contain only one set of chromosomes, that is 16. The number is 16. Whereas female is diploid. Queen and workers bees contain 32 chromosomes. That means this is the diploid state. And in honeybees, diploid state is observed in queen and worker bees, where the number of chromosomes is 32. Now, males produce sperms by mitosis. Males have no father, therefore, and cannot have sons, but they do have grandfathers and grandsons. Just this is just a fun. You can just remember that the males produce sperm by the process of mitosis, whereas in females, this condition of queen and workers is also decided dependent on which type of food they are taking. That means on which type of food they are taking. Females that feeds on royal jelly becomes the fertile queen, and other females which feed on honey and pollen become sterile, called as worker bees. If a queen bee mates with one drone, her daughters share three-fourths of their genes with each other, not half as in XY or ZW system, three-fourths. This is little different, just try to understand. So the female progeny shares between them 50% of the mother's gene, that is 16 out of 32, and 100% of the male parent's gene, entire male genome, that means 16 chromosome. So it accounts to 50, 100, but that means 150 out of possible 200, considering the male genome of 16 chromosomes as 100% of the male genes. Okay, so this calculation you don't remember, no need to remember, just remember that in case of honeybees, this is the queen diploid, this is the worker also diploid. The queen and worker, they are decided dependent, they are decided by the type of food they eat, and drone, that is the male. So male having only 16 number of chromosomes and the females with the 32 number of chromosomes, and this is what haplodiploid sex determination in bees. Queen is diploid, drone is haploid, and this drone which is shown here, that results in the diploid workers also. And when the drone mates with the queen, again, as I told you, sister and workers 75% relate, they are related to each other, sister workers. Just remember that haplodiploid state is observed in honeybees. I will stop here.